7.5 million students in the United States public school system are neurodivergent, and I'm one of them. Hello, my name is Abdullah Ali. I am autistic, and I'm a student in high school, and I have various stress, anxiety, and executive function challenges. These challenges made me interested to research and learn more about genetic causes and treatments for autism. Also, as a Muslim, I'm very interested in Islamic bioethical concerns about genetic treatments and how they're administered. So to just explain what is autism, is a autism spectrum disorder consists of various behaviors that are observable and can range from mild to severe. This can include social and also executive functioning challenges. For topics, I will explain about genetic and non-genetic causes, genetic and non-genetic treatments along with their risks and benefits, treatment concerns, and lastly, I will give a recap and I'll also explain about genetic treatments using an Islamic framework in the form of a bioethical discussion. So for genetic causes of autism, there are monogenic and polygenic causes. For monogenic, this is a single gene mutation that can occur, and this single gene mutation can drastically increase the chance of having autism. For example, the ANKRID11 and SHANK3 genes. If these genes are mutated, if even just one of them is mutated, this can impact neuronal communication. For polygenic causes, there are phenotypes that can be traced back to, but because one or more genes can be mutated that can increase the risk of autism, then there isn't a specific gene that can be identified. For example, the phenotype impacted microtubule structure and function in neuronal cells. This phenotype can't be traced to just one gene. And this is why, for example, either the MAP1A or MAP6 or possibly both genes can be mutated. For a non-genetic cause of autism, a main factor is an environmental cause, specifically maternal stress during pregnancy. And for this, lots of maternal stress can cause hormonal imbalances in the offspring. This can result in low testosterone and high cortisol levels. Cortisol is a stress hormone, and this can ultimately cause autistic individuals and children to have excess stress and for long periods of time. For monogenic treatments, there are three kinds: recombinant DNA-associated viruses, RNA knockdown, along with CRISPR. And the purpose of RAVs is to replace single gene mutations with foreign DNA. And the second treatment is RNA knockdown. And purpose of this is to decrease the amount of messenger RNA in cytoplasm that's translated into proteins. This mRNA can have mutations that ultimately can code for defective proteins. And then for the last treatment, which is CRISPR, is to edit mutations without replacing or silencing any genes. And number one, for limitations, a limitation which is common for all three treatments is toxic dosages. Such high dosages can result in non-targeted cells receiving too high and harmful dosages of that treatment. For RAVs, a unique limitation is possible immune system rejection of foreign DNA. And for CRISPR, delivery methods are an issue. For polygenic treatments, there are two main treatments: amino acid replacement, which is to correct a protein deficit by intra-CNS transduction, CNS being the central nervous system. And then the second treatment is fixing defective proteins to correct phenotypes. So for the first treatment, for the amino acid replacements, an example of this is in a research paper on mice, restoring the blood-brain barrier solid carrier transporter 7A5, and also correcting a deficit of branched-chain amino acids in the central nervous system. For the second type of treatment for defective proteins, this treatment is common in autistic individuals. This treatment also impacts the amygdala. It impacts mutations which affect the amygdala. The amygdala can impact social functioning and learning. An example of the defective proteins being fixed is the NLGN3 mutation, which causes synaptic plasticity challenges and conditions which are related to Fragile X syndrome. And once again, from this, either the FMRP gene or a different pathway, the P13K pathway, can be corrected with RNA knockdown because one of these two things is overexpressed, the FMRP gene or the P13K pathway. For pharmacological treatment, there are 4 medicines in 3 categories. The first category is antipsychotics, aripiprazole, and risperidone. And this helps to reduce anxiety and improve social impacts. However, the limitation is that these antipsychotics can cause weight gain. For CDPVB, this is to weaken neuronal connections caused by a defective mGluR5 neuronal receptor. However, additionally, this helps you reduce anxiety in autistic mouse models. For propranolol, this helps to reduce blood pressure and anxiety symptoms. This medication was also used in a research study on autistic children over the course of 3 months. And for this, although the high blood pressure was lowered and the anxiety decreased, the root causes of anxiety was not completely addressed. For non-pharmacological treatments, there were 2 kinds. Number 1, hyperbaric oxygen therapy and dietary and behavioral therapies. For hyperbaric oxygen therapy, the increased oxygen in the pressurized environment may help to improve social interactions and reduce neuroinflammation in the brain's cortex region. And however, the issue is that this treatment is still emerging. It's not completely developed yet. And for behavioral therapies, this consists of changing in an individual's environment to alter any behaviors that therapists hope to change or hope to improve. For dietary therapies, the objective is to increase an autistic individual's vitamin and mineral consumption. For examples of behavioral therapies, there's applied behavioral analysis, which is to treat many different skills. For cognitive behavior therapy, which is the second kind, which is also known as CBT, this is to reduce negative emotions. Benefit ABA is if young individuals receive this treatment, then many skills may improve. However, a setback is the initial uncertainty of phenotypical improvement, causing parents lots of stress. And for many different behavioral and even dietary therapies, if the autistic individual is not flexible to treatments, then there's no purpose for the treatment. For treatment concerns, there's the administration of treatments, which is if autistic individuals have phobias towards needles, then intra-CNS transduction has no purpose for being administered. Second of all, for concerns about ABA, if autistic individuals have to comply consistently because to ABA therapists, neurotypicals always comply, then this would result in an ethical concern of autistic individuals having to do things like a specific way and always do that and see their behaviors as being wrong rather than being different. And then for another concern, which is long-term treatment plans, because phenotypes may vary, then the type of treatment given, along with how long it's given, may vary for each autistic individual. This is related to treatments for coexisting disorders. For example, although treatments for behaviors that are associated with other disorders like ADD and ADHD may be fixed, their treatment plans should also be developed for treating behaviors associated with autism. And lastly, for access and affordability of treatments, one should be aware about medical insurance along with payment methods to cover such expenses with genetic treatments. And to recap what was covered, genetic cause of autism, like monogenic and polygenic mutations were covered, along with non-genetic causes of autism, which was mainly about maternal stress during pregnancy. Genetic treatments and non-genetic treatments, including pharmacological and non-pharmacological treatments, were covered. And treatment concerns were also discussed, like the administration, the concerns about ABA, along with access and affordability, and then treatments for long-term, along with treatments for coexisting disorders. And lastly, for the bioethical concerns, this is mainly for unnecessary gene editing for non-medical purposes. Islamically, there's wisdom behind God's creation, and genetic treatment should be safe, medically necessary, with one's consent, happen to body cells and not sex cells. Otherwise, future generations may not have a say, and ultimately, if they have gene editing. And thank you for listening to this presentation. I hope you found this valuable.